So I'm just going to list off a couple of the main kind of neurocognitive decline associated diseases, what we know now to be cognitive decline. The first one here, mild cognitive impairment. This is, a diagnosis of people who are just kind of starting to lose touch with normal behavior, having some memory issues, getting disoriented, not quite being able to do math like they used to, just mild cognitive impairment. It's just what it implies. And then that is, a precursor to the dementias, of which there are several, 4 main ones. And the one we're going to focus on today is Alzheimer's disease, AD, but also vascular dementias, Lewy body dementias, which also is Lewy bodies also are very influential in Parkinson's disease, and then frontotemporal. They're very minimal. We're not going to talk much about them today. And then Parkinson's disease as one of our top 2 devastating neurocognitive disorders that this country and this world is facing right now, and numbers are skyrocketing. So we're going to focus on that. And then some other things that we'll just hit in small amounts are Lou Gehrig's disease, amyotrophic lateral sclerosis, and then multiple sclerosis, and then Huntington's disease or Huntington's chorea, another movement-related disorder. So this is another slide I borrowed from Donnie. He and I work together a lot, so I like to network with what he's doing. He's doing great things. And this is a slide he made that I really like, and it's the cycle that leads to neurocognitive decline. And at the top here, you see stress. Over here, you see insulin resistance and the formation of ages, which we talked about. Endocrine deficiencies and imbalances, we went over that in detail. Chronic inflammation, which in the case of neurocognitive decline and Parkinson's and Alzheimer's, we're going to see amyloid beta, or Lewy bodies, those broken proteins, misfunctional structures that gum up the neurological space. Oxidative damage, and there's the breakdown of our mitochondria, that inability to insulate the free radicals that we're producing as we make energy. Pathological stress, all kinds of it. And we're going to talk about concussions as a stress. We're going to talk about physical damage, acute stresses, but then also just long-term pathological stress. And cardiovascular disease, we'll talk about tomorrow, it's another big factor for us, is this pathological stress. Really drives the body to break down. Heavy metals, especially Parkinson's, but also in Alzheimer's, and then cognitive impairment being the result. So this is this cycle. And with more cognitive impairment, there's more stress. You're more stressed out. Can't figure it out. Can't get where I'm going. Damn it. That really sucks, and now I'm pissed off about it. Now you're stressed even more. And the cycle gets itself going. So since we talked about adaptability, adaptation, we talked about stress, I want to talk about a concept that came up in some of the research I was doing, the concept of cognitive reserve. Some people can tolerate more neuropathology without cognitive and functional decline, therefore develop dementia more slowly than people without this type of brain reserve. Healthier lifestyles are associated with declining prevalence of cognitive impairment and dementia. So our brain, our neurological tissue, actually has a certain cognitive adaptive reserve. And that's a really important concept. And we're going to find that as we apply that adaptation rule, that allostasis that we talked about, that stress response, it's happening in our brains, and it's very directly connected to such things as these Alzheimer's and Parkinson's disease underlying factors. So anxiety and depression. Look at the numbers here. Over 10% of Americans are on antidepressant medications, which we're going to look at, and how they're helping us. Almost 20% suffer from some sort of anxiety. Concurrent with impaired sleep and impaired cognitive function. One of the most common forms of this is general anxiety disorder, right? These are the chronic worriers that have an inability to relax. You can never quite settle down. Something is always just not right. Something is making you uneasy, whether it's the airplane, whether it's your child and how close they are to the edge, whether it's your cat. It could be anything, but there's just a constant worry about something. Is it going to be okay? That thought, that driving force is underlying so much of what's happening for us that I just have to make it a really important piece of this puzzle. And the impaired sleep part, we have to get people sleeping well. And I'll tell you right now, benzodiazepines are not the answer. Sleep is something that you can't get from anything else. There are cases, again, I'm talking about emergency medicine. Someone just can't sleep. And I'll tell you right now, for me, there are 4 essential things we need to have in life. The top 4 things. Number 1, we got to breathe. Number 2, we got to drink water. What's number 3? Sleep. Way before food. The Guinness Book of World Records, the longest time somebody without sleep is like 11 days. I'm sure he's absolutely insane, but 11 days. People go without food for 2 months. Gotta sleep. So I talk about the social environment, just to kind of, I really hit this earlier about just isolation, social media, loneliness, and it's tied to depression. When you're not connected to that meaningful experience, that community of people that puts you in a meaningful place in life, what's the point? And if there's no point, then why do it? We need to have a point to be here, guys. 40 to 50% of Alzheimer's and Parkinson's disease patients have depression. Just going to start to pepper in some therapies here, just because it's time to do that. SAMe, right? Acetyldenylsilmethionine. This stuff is great for depression. Not in every single person, but it can be really helpful. Vitamins, the B vitamins, right? B12 and folic acid, lower homocysteine, depression, homocysteine, Parkinson's disease, levodopa, Parkinson's disease, homocysteine. They're all really closely tied. NADH, key cofactor in the manufacture of energy. If you don't have energy, it's hard to be feeling good and not be depressed. N-acetyltyrosine, we're going to hit this again. That's the precursor to your thyroid hormones, tyrosine. It's a precursor to your catecholamines. It's very, very helpful in depression. Fish oil and all kinds of fatty acids. And then gamma-linolenic acids like borage, primrose. In combination with botanical and adaptogenic formulas and indicated botanical extracts such as hypericum, they all work better. Are we all in agreement here that when you combine nutrients and botanical medicines together, you get a better result? Okay, good. Skip that part. Okay, so dementia, Alzheimer's, and Parkinson's disease. Just a little bit of, just some figures, right? Most common neurodegenerative disease in the world is Alzheimer's disease, projected to have 81 million people suffering from this disease by 2040. Parkinson's is second, somewhere around 7 to 14 million people right now. These are huge impacts for us. When someone has Alzheimer's disease, are they the only one suffering from Alzheimer's disease? No, who else suffers from? Everybody who cares for them. Everybody around them suffers from Alzheimer's disease. People who are in this field, caregivers for people who have severe dementia and Alzheimer's, they are kind of suffering. It's like you tell that person something and they don't remember what you told them. You told them something else. You didn't tell them that. What did you really tell them? Did you take your pill? Did you not take your pill? Did you go to the thing? No. Did you talk to that person? I don't know. It's confusing and hard. And this is like, where this number starts to get a lot bigger as far as the impact socially. And then think about the cost, the treatments. I mentioned one pharmaceutical drug right now to get through phase 3 or 4 clinical trials and get drug approval, $500 million to $2 billion. One drug. I want to talk about the word dementia, because it's one of those words, again, that we just sort of accept as like this, we just talk about dementia, but in Latin it means out of mind. The Diagnostic and Statistical Manual of Mental Disorders has stopped using the word, instead uses the phrase major neurocognitive disorders to get away from the idea that you're crazy. Dementia means you're out of mind. We don't want to call people crazy. So even though we're using this term, it's a term now, I think that we're going to evolve past it, get more, not politically correct, just socially correct. Just take care of each other better. 47 million people were living with dementia in 2015, triple by 2050. What are the comorbidities? Diabetes and hypertension. There you go, metabolic syndrome. Mixed dementias, vascular brain injury, strokes, concussions, microvascular infarcts, any kind of hypoxic events. 80% of dementias are in people aged 75 years or older. Remember we said what's the biggest risk factor for neurocognitive decline? Aging. Just to look at what's happening in high income versus low income, low and middle income countries. Look at this. this is our high-income countries. We've got a lot more resources. Everybody faces the same stresses. People around the world. Look at like Malawi right now. That whole region, the Congo, they just got hit. I mean, what was it? How many people was it? 750,000 people affected by that cyclone? Idai. Those people are suffering, man. That's huge stress. Risk factors, the well-defined ones are pretty common. We all can think of, diabetes, so blood sugar imbalance, and, all of our dietary problems, what that leads to, right? Midlife hypertension, obesity, physical inactivity, smoking, depression, social isolation, low educational attainment, not getting your brain put together in a complex way, hearing loss, very interesting one. This is a very interesting one, right? People who don't get hearing aids because they don't want hearing aids, they don't hear very well, and a lot of them then the world gets a little confounding because we use sound and sight as our primary communication signal systems, right? And when one of those starts to go away, and as we look back at that kind of early Chinese medicine idea about essence and the loss of it, the kidneys open into the ears in Chinese medicine. The channel goes into the ears. That's how you listen through your kidneys. And as that system starts to weaken, and as we can't hear well, you start to get mixed messages. You're confused. Did I hear that? Did I not hear that? And then you wonder, is it my brain or is it that I didn't hear it? And then you start to get into the self-questioning, self-doubt, and that can be a big part of dementia development. So, sometimes getting people on hearing aids is okay in this world today, just like we get glasses. How would I be without these? I don't know. It'd be a challenging world for me. Unclear factors. And I'm saying unclear because in the research, in the data, it's not 100% clear yet. Traumatic brain injury, concussion, we're going to hit that one hard. Sleep deprivation, proton pump inhibitors or H2Is, histamine blockers that reduce hydrochloric acid, benzodiazepines, they target the benzodiazepine receptor in the GABA region, and the use of selective serotonin receptor inhibitors. Are these possibly associated with dementia? What do you guys think? Maybe? Should we take a look? Okay, we'll get there. So what happens in dementia? Mood disorders, depression we already talked about, psychotic disorders, little things like stealing things, like taking things, not realizing that that's not socially accepted, that we've all agreed that we don't do those things. Inappropriate behaviors, aggression, purposeless activity. Like I said, let's get our guts happy. Let's make purpose, right? Purposeful experience, meaningful experience. Mealtime behaviors, sleep disorders. These are all things that are all part of the dementia picture. Kind of hit this one. Greatest risk factor is age. Most modern approaches in medicine are non-curative, focused on modifiable factors in lifestyle. That's kind of like when you look at these big dementia statistics and big dementia groups, the idea is that, if we exercise more, if we get, we surround ourselves with more people, we have more community. These are the kind of approaches, and I think those are all really good things, but without the botanical medicine piece, without the diet piece, without the nutrition piece, without the meditative practice piece, without, all the other things that we bring as doctors, they're not going to see the kind of results that we're going to get with people. So we're going to zero in on Alzheimer's disease from dementia. It's the most common form of dementia, right? 60 to 80% of cases of dementia have Alzheimer's disease. It's the 6th leading cause of death in the United States. It's a big deal. 13% of people over the age of 65, 45% of people over the age of 85. It's characterized by a progressive deterioration and death of neurons that gives rise to deficits in memory, language, and reasoning, and impairs daily living activities. So the modern understanding of this disease, Alzheimer's, is that there's this stuff called beta amyloid, amyloid beta protein, and this stuff is the problem. So most of our efforts have been focused on getting rid of this stuff. Lots and lots of money, lots and lots of resources, lots of drugs, trying to find a way to get rid of beta amyloid, amyloid beta, because it's building up in the brains. And when we look at the brains of people that died with Alzheimer's, we autopsy them, we compare them to the people who died without Alzheimer's, the people who had Alzheimer's have a lot more of this stuff in their brains. It's pretty much undeniable at this point. It's just, we know that this is associated. But this is not the cause of Alzheimer's disease. And so again, all the work we're doing on beta amyloid is not targeting the source of why the beta amyloid is there. But it is there, and it's a big factor. So what does it do? It accumulates, hardens into plaques, leads to neurotoxicity, disrupts nerve function, causes death of nerve cells, not good, leads to the formation of neurofibrillary tangles. That doesn't sound good. Brain should be relatively organized, right? The amyloid beta precursor protein, APP, is the source of A beta that accumulates in the Alzheimer's disease brain. All of this just says amyloid beta is a bad thing, and we find it in people who die with Alzheimer's. Make sense? Okay. Sort of. Maybe. Microscopic view. Here's an unhealthy cell. Here's a healthy cell. This is what we want. Here's some tangles. Here's some plaques. Starting to build up. There's material that shouldn't be there built up around the neurons that's inhibiting the flow of information. So the amyloid hypothesis is that the accumulation of amyloid beta in the brain is the primary influence driving Alzheimer's disease pathogenesis. The rest of the disease process, including neurofibrillary tangles, containing the Tau protein we'll talk about, it results from an imbalance between amyloid beta production and amyloid beta clearance. It's a focal point for pharmaceutical research. But as usual, those of us who are doctors of natural medicine of some kind, we're looking for the cause. We're looking for why are we building up amyloid beta? Why are there neurofibrillary tangles and Tau proteins that are over-phosphorylating? Why is this happening? And how do we prevent it? How do we change it? And modern medicine is going, holy crap, if we could just target this stuff and get rid of it and make people better, we could make trillions of dollars. I know, I'm kind of harsh. So amyloid beta is an antimicrobial peptide, right? It's not an independent cause. These three studies here, take a look at them if you want, but the idea that it's an antimicrobial peptide is that there are microbes that are imbalanced in the brain, and it used to be thought the brain didn't have microbes in it, right? It also was thought not long ago that the brain didn't have a lymphatic system, but we know now that there are lots of things going on in the brain that we didn't think were there. There's all kinds of viruses. Cytomegalovirus has a direct link with glioblastoma. There are now neurolymphatics, lymphatic networks in the brain that were just discovered. It was called the Bigfoot of neurology when it was found in 2016. The brain works in sync with our microbes. So one of these hypotheses is that the amyloid beta is actually produced by the body in response to microbes that are imbalanced due to changes in the ecosystem surrounding the neurons. It's the ecosystem that defines which bugs thrive in that space. If the wrong ecosystem is present due to the lifestyle choices that you've made, the wrong bugs are going to live there, and the result is that the body makes the wrong proteins. The infection hypothesis. The innate immune system dysregulation hypothesis. These are ideas. And what I say is that it's really about the complex organism hypothesis, which means there's a lot going on, and these are all part of a bigger picture. And yes, there are microbes that are present, and yes, they do have a role in amyloid beta production. But let's get back to the source again. We're not able to quench those free radicals. We're not able to regenerate the neurons with our anabolic hormones. We're not able to calm the inflammation with our essence, our steroids, our DHEA, our neurosteroid. And we're not able to maintain a microbe population that's most supportive of us. We aren't able to make the neurotransmitters that support clear communication among the neurons. All these things underlie this. This is a result. Some of the links here with viral, and this isn't one of your slides. I added this because I forgot to put it in the slideshow originally. Sorry. But it's here now, so I tell people to take a picture of it, or I can give you a copy of it. But one of these studies said that this is the most compelling link ever presented to Alzheimer's disease, is viral infection. And it's both herpes simplex virus 1 and human papillomavirus 6 and 7. These two, the HHV, were twice as present in people that died with Alzheimer's disease than they were in people who had normal brain function. Bacterial infection also implicated, again driving this idea, And if you wonder about this, was there really a bacterial link to neurocognitive function? The first place to go is syphilis. Syphilis was a disease that was sexually transmitted. You know, it still is, but it was much more prevalent 100 years ago. The spirochete that causes that disease leads to a condition called neurosyphilis, which is where there is damage in the neurons, and people have neurological decline and die from the disease. It's a very clear link between bacteria and neurocognitive decline. So these are all factors, but I want to say again, if we just start finding, oh, this virus is the problem, and we target that virus with a drug, are we going to fix the problem? Probably not. If we target the amyloid beta, are we going to fix the problem? Probably not. So this right here is super important to me, and I'm going to say this because it wraps into everything that's important in our life right now. You guys ever heard of Greta Thunberg? She's a 14-year-old girl from Sweden. She spoke at the United Nations Climate Change Conference. She spoke for 3 minutes. 14 years old, she decided, she's autistic. At 11, she decided that life wasn't worth it. She was so depressed, she went down, and then she recovered. She came out of it, and she got on a new mission, which was that she was going to change the world and make us realize that there's nothing more important than saving our ecosystem, our human ecosystem on this planet. And she's amazing. And her whole story is, one of the things she says is, hey, when I'm 75 years old, actually, she says, in the year 2075, I'll be 78 years old. And at that time, when I'm talking to my children, I wonder if they're going to ask me, how come people back in 2019 didn't do something when there was still time? When there was still time to change the way we do things before it's so broken down that we're dying and suffering without a solution until evolution takes place and we go through that process. Why didn't we do something then? And this is a great analogy because what we can do for people before they have amyloid beta built up in their brains, which is it's a material, it's a stuff, it's hard to get rid of it, before they have tangled neurons, before they've got all the preclinical stuff that goes into the development of the disease itself. If we can treat them before that, if we can curb how long it takes for them to develop these problems, if we can stop them from developing, we can be very potent healers. But it's really hard to change the physical structure of the brain once it's got all kinds of tangles and messes and amyloid and stuff in it. We can help, but we can do so much more now to help people not get here. So what are the neuropathological characteristics of Alzheimer's disease? Plaque formation, fibrous tangles, nerve cell death, mitochondrial impairment. I just keep hitting these things. Neuroendocrine decline, the dysfunction of that HPA axis, hypothalamic pituitary adrenal axis. Hormone disruption, inflammation, oxidative stress. What are the main pathways? Glutamatergia, we talked about that. Glutamate, overexcitation. Adrenergic, the beta-adrenergic receptors, like all over your heart and your nerves that stimulate processes. Serotonergic, dopaminergic, and cholinergic. Acetylcholine, that muscle memory neurotransmitter that conducts the bulk of our neurological energy across synapses in our brain and across our bodies. So let's hit oxidative stress for a minute. Talking about Alzheimer's disease. It's an imbalance between the reactive oxygen species and the antioxidants that we have in our bodies. And I don't like the word antioxidant, but it's redox cycling. Cycling things, helping those reactive species of molecules that have the capacity to damage other structures with their high-energy bonds, of high-energy molecules, quenching them, settling them down, making them not highly reactive, conjugating them with glutathione, conjugating them with thyroidoxin, superoxide dismutase. All these cool compounds we think about, they quench the free radicals. When those are weakened, the mitochondria are impaired, our energy system breaks down, our repair systems fall apart, and we're not able to heal. I've been talking a lot about inflammation and excitation. So inflammation in the brain leads to an overactivation of neurons. What's important here is that too little activation means the signal is too low, too much means the noise is too high, and either way you will lose cognition. Again, we want to find that midpoint. Inflammation is essential, but inflammation has to be controlled. It has to be calmed back down. as we'll see on Sunday in my talk on pain and inflammation, inflammation can quickly get out of control without the right mechanisms to keep it in check. So what happens when we have over or imbalanced neuroinflammation? When we have that excitation happening, neurogenesis, that repair system, isn't there anymore. Again, when energy goes into action, there isn't energy for repair and regeneration. When energy goes into rest and sleep, there isn't energy for action. That's a perfect little balance for us, and we keep that every day. So the mitochondria, this is so important, right? Significant role in both Alzheimer's and Parkinson's, neural damage, death of neurons. How do we address it? We've got to address the endocrine system as a whole, address the mitochondria, help them convert nutrients and energy, quench and insulate that process, and enhance the mitochondrial membrane integrity and fluidity, and also the potential, the electromagnetic potential across those membranes. That's where all this exchange is happening, really elegant exchange of energy manufacture, the generation of ATP, right? The Krebs cycle, oxidative phosphorylation, and the electron transport chain. You guys all know this from biology, right? It's a beautiful system. It's so elegant. And you watch what cancer does. Cancer does glycolysis. Super inefficient. Just like what we're doing in the world. Just, let's grab all the fossil fuels, we'll burn them. Woo! We can do it. Very inefficient. Huge waste, huge byproduct, huge cost. But it's easy to get and it's there. So let's talk about Alzheimer's and genetics. A lot of genetics associated with Alzheimer's. Amyloid-related genes, 60 to 80% of AD. Familial AD with onset less than 60 years old, only 5%. And this is a really, this presenilins are these interesting little gene functions on chromosome 14 and 1 that have a role with these things called gamma and beta secretases that basically cut the beta amyloid into little sections, and those sections become much more damaging. When this process is going on, when you have the wrong presenilin genes, the ones that are mutated, the beta amyloid that you manufacture doesn't get cleared. It gets stuck. These people are at high risk of Alzheimer's disease. But most of what we're going to see is non-familial, later onset, again, age-related Alzheimer's disease. And apolipoprotein E, and of course E4, epsilon 4, is the main one that we've been working on. There's a lot of other genes associated, but we're going to focus on apolipoproteins because they're really important. They're cholesterol transport molecules in the brain. And I just want to mention, cholesterol is so important for us, so important for neurocognitive function. It's the backbone for building testosterone and estrogen, for our anabolic hormones. It is how we transport and use energy in our brains. It's how we insulate our brains. We need it. So the fact that we have this huge market of blockbuster drugs to just lower cholesterol as a target again, it's inhibiting us from becoming what we want to be, which is healthy aging adults. Again, urgent, emergent situations, these drugs are great, but in this case, lowering cholesterol can be very damaging. In people that have the apolipoprotein E4 genetic alteration, it doesn't function how it's supposed to. And what it's supposed to do is it's supposed to repair the nerves and remodel them by bringing cholesterol into the cells, into the neurons. And when we have injuries and stress, apolipoprotein E is a big response. Just like I said that the beta amyloid was a response to infection, right, in the brain. So like maybe an antibiotic kind of body peptide. But in the case of APOE, it's a very important lipoprotein that we can analyze in the blood and understand in people. And if it's not functioning well, if it doesn't do its job, we're not able to repair and remodel our nerves. So there's three main isoforms. The E4, epsilon 4, is the most strongly connected to Alzheimer's disease. 10 to 15% of the population. You can test this in all your patients. We can all get this. It's a part of like 23andMe. You can get this really easily, find out this information. And if people have this, it's about 10 to 15% of people that you're going to see in general. It's a very strong risk factor for Alzheimer's disease because this E4 apolipoprotein doesn't do that job of bringing the cholesterol into the cells and clearing the beta amyloid as it should. It's dysfunctional. Okay? E2 is actually neuroprotective. It's only about 7% of people that have that. But the people that do have that have a little bit of neuroprotection. And then E3, which is about 75% of people, those are normal people. They have like the neutral sort of relationship to the formation of amyloid beta. Does that make sense? Apolipoprotein E4, the bad one, so to say. It's the one that is associated with Alzheimer's disease. We're going to find out that, again, it's not the problem necessarily. It's tied to vitamin D levels. It's tied to all kinds of interesting things that we're discovering. But as a single risk factor, as an, out in its own vacuum, its own silo, it's very clearly associated. It results in truncated fragments caused by stress or injury. It increases Dow protein hyperphosphorylation. And the Dow protein is a microtubule stabilizer, right? Microtubules as a part of the cytoskeleton structure of the nerves and the nerve rebuilding process. When these Dow proteins hyperphosphorylate, this causes a breakdown in our stabilization, and then we get these neurofibrillary degeneration processes that get in motion. See, this is a slide I threw in at the end. See, neuro, I spelled it wrong. Cytoskeletal disruption, mitochondrial dysfunction, inefficient cholesterol transport, and then the overall result, a loss in cortical thickness and hippocampal volume in people that have this APOE epsilon 4 mutation. So we're back to this clearance idea. Accumulation and aggregation. When we can't clear the amyloid beta, once it's being formed, let's say it's in response to inflammation, it's in response to infection, whatever we talked about, right? It's being built. We need to get rid of it. It has a role, but if it sticks around, it's going to lead to problems. And if we can get rid of it, that's better. But with this APOE4, we can't get rid of it. It sticks around. And one of the big connections is depression. And it's a cardiovascular disease risk factor. So when people also have atherosclerosis, peripheral vascular disease, diabetes, it's going to amplify its effect. And the implications are bigger for mortality and for well-being in people, for quality of life. Here's just an overview. Decreased synaptic function. These are people with APOE4. You see real simple there. All the good things on the right are in decline. Loss of function. All those things on the left is a gain of toxic function. Atrophy of the brain. Remember, loss of that cortical mass. More tangles. Aberrant brain activity. More amyloid beta. So how do we deal with it? Again, peppering here some nutrients. Vitamin E, N-acetylcysteine, and alpha-lipoic acid, all considered antioxidants. When you combine them with phytonutrients, things like green tea and resveratrol, combine it with curcumin, you get a reduce in Alzheimer's disease even in the presence of APOE4. So these are things that, like I said, it's harder to deal with it once it's formed. It doesn't mean that we can't help those people. It just means that we're going to get better results if we can get it before it's formed. But these things will also help to eliminate what's there. NAC plus acetyl L-carnitine, enhanced cognitive function in APOE4 positive genotypes. And in mice, improved cognitive function. So there's just some of the things that are, with modern research, just found to be directly affecting people with APOE4 and improving their cognition and reducing their breakdown. So some of the blood markers that we like to see that are associated with this, DHEA sulfate, I mentioned this, it's so important. DHEA sulfate is the one to check in the blood because it's much more stable than DHEA, which can fluctuate a lot. So measure DHEA sulfate. Insulin, fasting glucose, we talked a lot about this, the metabolic syndrome, estrogen, testosterone, apolipoprotein E4, homocysteine, inflammation, C-reactive protein, and then interleukin-6 and tumor necrosis factor alpha. These are big drivers for inflammation and immunological activity. Oxidative stress, you can measure glutathione. This is a really important, part, but most people are going to be deficient. They're like the Nutri-Eval test from Genova is a good one, so you can see the kind of what's the antioxidant status. It's important things to look at. Most people are going to need help in this area, whether you look at it or not, but sometimes it's nice to show people the numbers. Say, hey, this is what's going on in your body. This is where you're deficient. Secretory phospholipase A2, 2A. This is the plaque test, and I'm going to hit this one hard tomorrow morning. It's another marker associated with inflammation and in the neurological system and also in the cardiovascular system. And then alpha-1 anti-chymotrypsin. There's another protein that are more directly related to neurocognitive decline. I'm going to go quickly into those. But the two things, estrogen, older women with low estradiol are more likely to experience a decline in global cognitive function and verbal memory. Estrogen is important. Testosterone deficiency is associated with multiple deregulations that include depressed cognitive ability and depression. We hit those anabolic hormones and reproductive hormones really well earlier today, so I'm not going to spend too much time on it. But homocysteine, I mentioned, can be very highly toxic. It's an inflammatory compound in the body that's produced through our regular methionine cycle. I want to get it. We'll look at this in a little more detail. But homocysteine being around increases your risk of Alzheimer's about 200%, linked to depression, Parkinson's, Alzheimer's, and neurological disease, an independent predictor of the development of dementia and AD. And this is something that we can look at in the blood easily, and we can control this easily. I can't tell you how many patients I have seen them have elevated homocysteine levels and have got them into the range that we're looking at of under 9. It's not hard to do. It's successful in almost every single patient, and it's so helpful. Yet it's not run by your modern medical doctor. It's not even run by most cardiologists in the lab reports that I see. One thing we're going to hit on with Parkinson's disease is when we get to levodopa, the primary treatment for Parkinson's disease is that it actually drives up homocysteine. So we got another problem here. You're making a solution that causes another problem that we don't have a solution for. Well, we do, but in general, it's not treated. Increased risk of bone fractures, peripheral neuronal dysfunction, and Parkinson's disease, and there's that target under 9. So what do? We need methylation agents for efficient conversion. Folic acid, B12, and B6, the primary methylators that are going to help with homocysteine metabolism, so we can keep levels low, so it's not circulating in the blood, driving inflammation, causing more damage. Choline from lecithin and betaine from beets, trimethylglycine. These are methylating agents that can also lower homocysteine. I find that they're actually really important in a lot of people. Some key nutrients, creatine, riboflavin, zinc, magnesium, all also important. The main thing you'll see in the research is on folate, folic acid. And of course, we talked about this SNP when I asked you guys what's the most common SNP associated with depression, MTHFR. When people have MTHFR alterations, especially in the 677 domain, that area specifically will lead to increased homocysteine and increased cardiovascular risk. This one here, this is MTR. These are associated with B12 recycling. So if people have issues here, B12 is very, very helpful. And then this is the cystathione beta synthase, which I'm going to hit on the next slide. So here's just a little, for me, this is a little thing now. It used to be big when I first saw it, but now it's really small. If you haven't seen it, it's probably complicated and weird. And I won't beleaguer this point, but here's homocysteine. This cycle right here, the methionine cycle, methionine comes from protein in our diet, especially in meat and nuts, seeds, really rich in methionine. And then methionine goes through this process of being metabolized. There's S-adenosyl methionine, right? This goes into building RNA and DNA and proteins, how we convert protein into usable stuff for us. That's the methionine cycle. Homocysteine is a product of this process and a natural one, but it should be in balance. This cycle has to drive in a circle to make homocysteine go past this process and become other things. In order for that to happen, this cycle, the folic acid cycle, this has to work also. Here's MTHFR. This is where your folic acid comes into play. Here's B12, this is that MTR pathways right here. They're both important to drive this cycle, which is going to drive this cycle, to make homocysteine move through this process of metabolism. Now, I just mentioned something called CBS, the cystathione beta synthase, this guy. That is an altered gene in a lot of people, and it's associated with sulfur problems, people who can't do sulfur foods. You've seen that. People are like, I need a non-sulfur diet, like things like Brussels sprouts and broccoli and cabbage and anything like that that has lots of sulfur. Well, they'll get, really tough effects. They'll get, heart palpitations, insomnia, rashes, whatever. Those people have oftentimes what's called an alteration of the CBS gene. And in that case, that gene, when it gets altered, it goes into metabolism. So altered gene function means that this becomes a dump. Vroom. Homocysteine starts going really fast down here. And there's a side pathway that you don't show here, that where the cystathione doesn't get moved all the way down to cysteine and then glutathione, which is our primary quenching molecule, right, that we use to quench all the free radicals. It goes over here and it makes taurine, ammonia, and other sulfur compounds called sulfites and sulfides, hydrogen sulfide. That's bad. And so when people have an alteration here, it goes over this way and they start getting all these byproducts that result in toxicity around sulfur, which sucks because you want sulfur because it's what makes glutathione. So a little key here is molybdenum. If people have that and they want to eat sulfur, molybdenum really helps. You know, a little bit, like 200 micrograms. It's a really good amount to use for people that want to get through that. But the idea here is that you can facilitate homocysteine through choline, like I said, from lecithin or from eggs, betaine from beets, this turns this cycle, B12 pushes this cycle, folic acid pushes it here, and all those things are connected to metabolizing homocysteine and getting it out of the blood so it's not toxic. You guys doing okay? Good. Overload? No? Good. Okay. So just a couple of these specific markers, interleukin 6 and D-dimer. D-dimer is a peptide that's cleaved every time a clot is broken down by the body. It's mostly used in the emergency room for, detecting thrombotic events, but it's a really good indicator of coagulation. And because coagulation and inflammation are tied together, it's really good to see this. It's a risk factor for, of course, disease, cardiovascular disease. And when you combine it with C-reactive protein and interleukin 6, which is a main driver of the inflammatory immune response, you see what's going on in people's bodies as far as, again, that fire that can rage out of control that leads to damage. Secretory phospholipase, inflammatory protein, the inferior temporal gyrus, this little part of the brain here in the temporal region, kind of down low by the ear inside here. This is where we see there's a lot of this, it's called plaque, the plaque test, but this is found in that area, and it's associated with a lot of amyloid plaque. So it's one of those inflammatory drivers of amyloid beta buildup that leads to cognitive decline, and especially Alzheimer's, right? And then this other one I put up here, these are tests that you can run. These are good things. And this, like I said, the plaque test we'll hit tomorrow because it's really important in cardiovascular disease. It's kind of like breaking through the idea that we're just going to study cholesterol, and hypertension. There's so much more going on. Alpha-1 anti-chymotrypsin induces Dow protein overphosphorylation. Like I said, that destabilizes the microtubules, breaks up the cytoskeleton, and leads to more tangles and beta amyloid buildup. And the interesting thing about this test is that it can be a biomarker for people if we want to discover, we want to say, are you having mild cognitive impairment? Are you having problems with memory and learning and spatial relations and processing? Those are all symptoms, but even before that, you might find that this is a little elevated in a patient, and you can control it by giving them botanicals that control inflammation. We need early diagnosis. We need markers that are there for us before you've already got enough damage that you're seeing overt clinical symptoms. So what are other important factors? Thyroid problems, heavy metals, concussion and traumatic brain injury, and drug toxicity. These are all big factors in what's driving this. The thyroid-adrenal axis, right? We talked about the HPA axis. If you want to get really down to what that system is, it's the hypothalamic, pineal, pituitary, thyroid, adrenal axis. All of those glands that are on the central midline of our body that regulate function of the organs. So the thyroid and the adrenal are key components to how people feel. So well-being, depression. And then the thyroid directs the metabolic activity of every cell in your body. It tells the cells to make energy, make ATP. So when that's not working right, people don't feel well. And then your cognition starts to decline. And what underlies all this is adrenal fatigue, is chronic stress leading to a maladaptation, to an overproduction of glucocorticoids, and a breakdown of the kidney-adrenal axis and the thyroid-adrenal axis. When that falls apart, neuroendocrine stability falls apart, and we end up with lots of neurocognitive decline. It's much more rapid. So we can address this by getting people's thyroids back together, and it's not just giving them thyroid hormone. It's actually working on what's the cause. Why isn't their thyroid making enough? Going back to the feedback loops, getting those adrenals working well, getting their stress levels reduced, controlling the inflammation, oxidative damage, on and on. Heavy metals and minerals. Copper and iron, two of the big ones. We've got copper pipes. Interesting that in Rome they had iron pipes. Copper is one of those interesting nutrients that we need for a lot of things, but it can also be problematic. It can accumulate. And iron, of course, with ferritin, can be an acute phase reactant involved in inflammation. So when either of these things are too high in the body, you get problems. More cognitive impairment is associated with copper and iron. Aluminum, obviously, we know it's just not a good thing to get in our bodies. More accumulation of amyloid associated with copper and aluminum. And also the progression of neurodegenerative diseases with copper. So I just want to hit these because they're all really important. And, if you're looking in a naturopathic way at people's accumulation of metals, these are some of the big ones that you're going to find in folks that have Alzheimer's disease. Calcium also, we're going to talk about calcium tomorrow also, because taking a lot of calcium we've been doing for a long time, and it's just not helping, and it actually has problems. There's a lot more to calcium than we think. We are basically banks of calcium. Our bones are banks of calcium. And it's not about having not enough of it. It's about not getting it to go where it's supposed to go. It shouldn't be in our bloodstream in high amounts and in our bones as bone spurs. It shouldn't be reducing our hydrochloric acid production. It shouldn't be resulting in atherosclerotic calcifications. That's too much calcium in the wrong place. So I want to hit concussion and traumatic brain injury. One of the most remarkable things about this is that, there's been all this energy lately in the sports, contact sports areas, boxing and football and things like that, because all these guys, as they're getting older, they're not that old before they start having dementia and Alzheimer's and Parkinson's and neurocognitive decline. They're like, what's the connection? The connection is concussions. It's getting hit in the brain, getting hit in the head, and how that damage accumulates. Rob?
Do you send your patients to naturopathic doctors or holistic doctors to do EDTA challenges? I'm just curious.
So Robert is asking about EDTA challenges, which is, basically a chelating agent for heavy metals. And I would say that, I think it's absolutely something that's worth doing with patients. I'm much more of a botanical medicine guy. My focus is on helping the body through the NRF2 agonists to build glutathione and then providing the body with all the substrates to try and, get that process happening. And I've used a lot of the Quicksilver stuff, which is Chris Shade's company, that he uses a lot of oral glutathione, and then also like phase 3 detox stuff, like using clays and charcoals to help bind up metals once they've been pulled into the bloodstream and then they're exposed to the intestine. There's a whole bunch of ways to go about getting the metals cleared, but the EDTA challenge is a good way. I think the way that I've normally sent them to naturopaths for is a DMSA, DMPS challenge. A little bit milder, but yeah.
Do you see any information with the NAC's ability to pull out metals?
Yeah.
Post challenges or whatever it might be?
Yeah, exactly. And, and the thing is, NAC is, it's one of the main building blocks of glutathione, so it's so essential in that way. And the N-acetylcysteine has that acetyl group attached to it, which gets it through the blood-brain barrier. So it helps it provide that substrate in the brain to make glutathione locally to quench free radicals and get rid of the heavy metals that way. So what's the question specifically?
Yeah, I wasn't even thinking about it. I was just thinking talk about it as a chelator, but maybe that's what they're just saying.
Yeah, it's a precursor. Yeah, it's a building block because glycine, glutamine, and cysteine make up glutathione, and the body needs enzymes, the NRF2 enzymes, to build those glutathione molecules. So you put in the plants that direct the enzymes, you put in the nutrients that are the building blocks, and then the body starts making it. That's my way to go about it. Again, I'm back to that. How do I get the body to do what I want it to do instead of giving it what I want it to have? So concussions. The coolest thing I was going to say about this is that even in mild traumatic brain injuries that do not result in loss of consciousness, there are long-term neurodegenerative consequences. So the risky thing about this is that people don't even know they had concussions. You just fall. You didn't know. I didn't lose consciousness. Just hurt my head really bad. A bunch of those over time, look at the risk factor here. These are in veterans that were studied here. Concussion without loss of consciousness led to 2.36 times the risk for dementia. Huge impact. Concussion leading to more serious, moderate to severe traumatic brain injury led to almost 4 times the risk of dementia. These are lifetime events that happen at some point along the way where your body responds to that traumatic event. Remember I said amyloid beta produced in response to a traumatic event, whether it's a microbe, whether it's an inflammatory insult, whether it's an oxidative damage overproduction. This is a major trauma. This is a stress that induces a response in the brain that leads to the structural changes that we see with these diseases. So I want to talk about the endocannabinoid system here, because this is a system that's getting a ton of publicity right now. And I mean, I know that CBD and THC, it's in the limelight right now as a medicine, and there's always something in the limelight. It doesn't mean that the thing it doesn't work because it's in the limelight. It just means that there's a big focus on it right now, and after a while it'll fade back and it'll be just another one of our tools. But right now there's so much focus on it that it's kind of fun to see the research coming out. And the research studies are starting to get into humans, but we've had so much, prohibition in being able to study this stuff that we don't have much there. We mostly have animal studies. But the ECS is part of this compensatory and repair system. And so we have our own endogenous cannabinoids that we make, right? We make primarily 2-AG, 2-arachidonyl glycerol, and anandamide. Those are our two main endocannabinoids that we produce in the brain to stimulate our cannabinoid receptors to change the way that things function. And it's mostly around protection and around memory and around appetite and around mood. Those are the places that cannabinoids work in our body. So we make them to induce those changes. What we've noticed is that under these conditions of traumatic brain injury, the body makes more endogenous cannabinoids. It makes them. There's higher levels when there is stress and injury. So we know they're protective. They have some role to play. Anandamide activates this TRPV1 receptor, which regulates body temperature. That's also the target of like capsaicin, pain-related nociception. Picks up that spicy flavor on your tongue, TRPV receptors, the vanilloid receptors. Reducing brain damage. This is with TBI in animals so far. Reduces brain edema, better clinical recovery, reduces the volume of infarcts, neuroprotection, tons of data on that in animals. And the CB2 receptor, which is all over the neurological system, has been associated with rebuilding nerves in people who have had strokes. Some new studies coming out. Remember, I said the studies on this there's going to be a wave of studies coming out on cannabinoids and their role in neurocognition and neurological decline. But until we get past the regulatory hurdles, we're still going to be using animal studies, which is what we have for most of our plants in that same regard. But here's the things that we know so far. Limiting the size of damaged tissue. And again, like I said, the discovery was that, wow, when the brain gets injured, when there's a trauma, there's tons of more endocannabinoid there. Well, what if we gave cannabinoids from a plant that makes them? Molecules that are very similar. We see that there's positive effects. Here's a new study. This is kind of the idea of where this is going. This summer, 100 former pro hockey players are going to be studied using CBD as a way to ameliorate their TBI-related symptoms, headaches, PTSD, emotional instability, anxiety, depression, and pain, all of which are precursors towards, neurocognitive decline. So we'll see what happens. But these kind of studies are going to be really interesting for us and help us to understand how this medicine can be part of our botanical toolbox as doctors of natural medicine. So if you're in the CBD realm, dose it in you have it in fatty acids. Buccal absorption is the best way to get into the body to bypass all the metabolic destruction that happens through the GI tract. There's only about 7% uptake of CBD and cannabinoids in the GI tract. So getting it in as a suppository or through the oral buccal route is much better, higher absorption. Most preparations now are, coming in an oil with the CBD as an extracted oil, the two together. And I'll just say one thing about CBD products is that it's this, just like with everything, the market is so focused on it that everybody's out to make a buck on CBD. So there are very few companies that are actually following through with the guidelines for the regulatory guidelines for how to make a GMP supplement or a GMP pharmaceutical. And that's what needs to happen in this regard. And it's not about isolated CBD, again, like we've been talking about all of our plants, and we will talk about. It's about the matrix of compounds that occur in the cannabis plant, as the hemp plant, and the matrix of those compounds, and with a focus on the CBD as being sort of a standardized part of it, that we understand that there's a lot of this active component. But you want the whole material together. So if you're going to explore this with your patients, just doing diligence on where their sourcing is really important, because people are starting to spike products with CBD isolate that's been isolated from who knows where the CBD material came from. People are trying to make a lot of profit on it. There's new technology now that, just like with everything, yeasts can produce CBD, so now it's starting to make it out of a yeast, but it's not the same thing. When a yeast makes it, then if it's made in a cannabis plant, it's filled with terpenes and flavonoids and chlorophyll and lignans that come with it. So if you're doing dosing with CBD, the starting dose generally about 10 to 20 milligrams, 3 to 4 times a day. Upper end of dosing, 300 to 500 milligrams a day. Main symptoms, side effects would be sedation. Once in a while, GI stuff with people, but mostly it's just sedation. People don't get high off of CBD. Most products out there have a less than 0.3% THC. That's what makes it hemp. So that less than 0.3% THC is actually an important synergistic relationship to the CBD and having a neurological effects. So there are good products out there. Yes?
So your dosing is the milligrams of CBD in the
Correct.
Not like hemp oil.
Correct. Yeah, so you want to have a product that has standardized to how much CBD is in it so you can understand. Any questions on this one? I just wanted to talk about it because it's a hot topic and it's going to be really important. We're going to find that CBD is going to help our brains.
My question is, does the body build up a tolerance? I've noticed with...
Yes.
And do you recommend changing the brand or the type so that it becomes more effective?
Well, what happens is that the endocannabinoid receptor network is a system that builds its receptor response based on how much of the actual ligand, right, the CBD it's facing or the THC it's encountering. So if you start putting more in there, then the body builds more receptors. So in order to get those receptors to all respond, you have to give it more material. It's just like the opioid receptor system or the nicotinic receptor system. They do the same thing. That's why people start smoking, and you get to that point, you're like, I had a little bit of tobacco first made me feel crazy, and then after a while, I don't feel anything. It's something like that will happen, but there's also sort of a modulatory effect where you kind of get to a level where the body, you don't really see any more tolerance. It sort of gets to a place where you've achieved some kind of, I don't know what it is, like priming of the endocannabinoid receptor system or something, that it sort of starts to be stable, like symptoms are managed. I haven't seen it fail to work in patients with epilepsy or other disorders. It generally, once you find a dose that works for people, like you can stay on it. With everything, I try to make it a little pulsatile. I don't want to just give somebody anything just every day, all day. Try to take it on and off, reduce dosing, change dosing, take a week off. Unless they have, a seizure disorder or something like that, where you just need to get it in every day, and then you'll just get to a stable dosing. And I haven't seen that dosing fail at some point.
Just notice with people with anxiety, like if you change the brand or the type of CBD that they use,
They may or may not get effects. Yeah, she's just saying that with anxiety, for example, which I would say CBD is not, that the 4 things would be anxiety, insomnia, some depression, and inflammation are the primary place, and pain. Those are the places that it targets. Question? Doug?
Just wondering if you could repeat the question.
Yeah, that's what I was going to do. The question was around tolerance to CBD, and then also around if you're using it for somebody who had anxiety. Sometimes if you change brands or change the potency or something, then it doesn't work as well. And so what's going on there? And I think it's important to understand that, like I said, the brands, there was a recent study that went out. There was a group that went out and they pulled, I'm trying to remember how many products it was off the shelf. But basically, the range was a couple of products had close to what they said in there, but a lot of the products had very little of what they said was in there, and a good number of them had none at all. And there were some weird things like people taking gummy bears and like injecting them with CBD and like just all kinds of like, just this. You got to, just don't just say, hey, go out and get some CBD. You need to go out and say, hey, there are some companies that are doing this within regulatory control, so somebody has oversight on the process, and they're doing it in a way that represents, a phytopharmaceutical product.
You may have answered with this tolerance, but I sometimes find people getting tired from.
Yeah.
Yeah. So I'm just thinking 3-4 times a day. I mean,
Yeah. Exactly. Some people won't find that, and so there's the Ethan Russo, one of the big researchers in cannabinoids. He was saying that there's a dose response curve where like at the lower doses people will get a stimulatory effect from CBD, and then at higher doses they'll get a more sedating. But then the ECS is so different from one patient to the next that one person can take 50 milligrams and not feel a thing. One person takes 5 milligrams and they're like, oh man, I feel like I'm so like just out of it. So yeah, well with cancer patients, for me, my target's 500. So but it's also with if people want to do cannabinoid stuff, you got to combine the whole cannabinoid structure. It's a whole process. But there are things we can do in that realm. Okay. Drugs for cognition, pharmacy credits for NDs. We're getting there. So here's the only approved drug treatments are for Alzheimer's disease dementia with Lewy bodies or Parkinson's disease dementia. They target abnormalities as a consequence of neuronal loss, but they do not modify the underlying neuropathology. And this is a quote, the Lancet Commission, right? December 2017, no drug has shown neuroprotective potential in humans. How many of our plants do you think are neuroprotective, have shown neuroprotective actions in animals? Tons of them. Tons of them. So what about using drugs for Alzheimer's? Is that a good idea? Well, they just act on symptoms, not really able to modify the disease effects. No evidence that any drug interventions delay conversion to dementia in a general population with mild cognitive impairment. So you're not slowing the disease, it's progress. And some of these drugs, have a mild effect, modest at best. But there are costs as well. And the drugs that I'm talking about right now for Alzheimer's are all drugs that are basically acetylcholinesterase inhibitors or anti-acetylcholinesterase drugs. And we'll talk about that because I've got a slide on that. Two more things here. There is a new monoclonal antibody, just to see how far this is going, right? Going after the beta amyloid. So now we've found that there's actually a monoclonal antibody, a molecule that will stick to beta amyloid to get the body to break it down and get it out of the brain. Immunotherapies are very interesting. They're very powerful, and we're going to have a panel to talk about them in cancer tomorrow night with Kenneth Profok and Donnie and myself. Because immunotherapy is a really amazing frontier, in treatment of many disorders. But again, there's a huge cost associated with this. The development of autoimmune disease is a big cost. But there may be a way that, as we've seen with cancer cases, that we can combine small amounts of immunotherapies with botanical medicine to create situations where side effects are minimal and the effects are positive, and we can use these things in severe cases. And this is an example, this adalimumab. This is one that does have some, possibility. And this may be an avenue that we're going to see some purchase. But again, it's targeting the beta amyloid, not the cause. And NSAIDs, as a direct, trying to block inflammation, they don't work. So what do the cholinesterase inhibitors do? They stop the breakdown of acetylcholine. Acetylcholine is broken down by an enzyme after it goes from one synapse to from one nerve to the other across the synapse. It gets denatured, broken down by an acetylcholinesterase, and that ACE breaks it down. So these drugs target that and say, well, stop breaking down the acetylcholine so that it can keep stimulating the receptors on the other side and give that signal transduction more purchase so that you can get more memory and learning and more connectivity. That's the idea. But one problem is that people who take these drugs have higher rate of mortality. They die sooner. They have a lot of side effects. Withdrawals don't work. You have to stay on it. Once you're on it, you're pretty much stuck on it. Because if you take it off, you'll just slide right back to where you were when you started it. Again, there might be some way that we can intervene with botanicals and help these drugs work better. And I've seen some of that. But I really don't like my patients on these drugs. So what are the ones that are in routine use? Donepezil, rivastigmine, and galantamine. Minimal clinically important difference on cognition in all the studies. Very little. Again, it comes back to that question, like with drugs that are have such a small benefit and are associated with a lot of side effects and cost, this is the best we can do? And we're approving these drugs? The differences are not clinically significant in behavior. And I mentioned, remember I got that slide with behavioral symptoms. It doesn't really change those in people. It helps them a little bit. They're not recommended for the vascular or frontotemporal dementias. And there's one other drug here, memantine, which is a drug that came out a little while ago for targeting the NMDA receptor. This is the kind of target of ketamine. NMDA has a lot of interesting effects. Memantine is associated with increased blood clots, some psychosis, and heart failure. One of the first drugs that came out as an approved cholinesterase inhibitor was Tacrin. And it was pulled off the market in 2013 due to it not working and it causing lots of sickness in people. These are second-generation drugs, Aricept, Razidine, and Exelon. And you can see what they're approved to treat. Mild to moderate, not approved for mild cognitive impairment, just poor outcomes in the studies. Aricept is approved to treat all stages of the disease, but again, it's not going to give much benefit. It's a small, modest benefit. And what's interesting is that if you put patients on these drugs, they get some cognitive benefit. Their learning and memory gets a little better. They're able to, face life, follow their map a little better. If you take them off the drug, they're right back to where they were or further below that. The NMDA receptor is a really interesting one. It's involved with glutamate. It's a glutamate receptor. So glutamate, that neurotoxin, that overexcitation process, it's an influx of calcium ions, which happens in a lot of receptors are, channels for ions to flow into the cell to change the polarity to have an effect. This NMDA, when it gets stimulated, calcium ions pour into the cell and create this excited state. By blocking that NMDA receptor, you would, the rational conclusion is that you would reduce the overexcitation and therefore reduce the excitotoxicity. Again, modest effect at best, no effects in earlier stages, or any inhibition of disease progression. So even if you're getting a modest benefit, you're still declining. So I'm going to throw another plant at us here. This is a Chinese herb, the Huperzia moss, Huperzia serrata, used in Chinese medicine for thousands of years. Swellings, fever, blood disorders, menstrual cramps. Wonderful plant with lots of benefit. There's a compound in this plant called huperzine A. This is what it looks like. And it's been defined as an acetylcholinesterase inhibitor. And it's quite effective. It enhances the cholinergic activity in the body. And when I say enhances cholinergic activity, I mean that when acetylcholine is moving from one neuron to the next, that compound, that huperzine A, helps facilitate more connection between those two neurons across that neurotransmitter. Connects those two neurons so it can make that signal continue onward. Okay, makes sense? So it's a neuroprotective substance. It penetrates the blood-brain barrier, has better oral bioavailability than the drugs that we just talked about, a longer duration than Tacrin, donepezil, and rivastigmine. And also, it does more than just target acetylcholine esterase. It does more than just that. Super safe, no unexpected toxicity. Compared to Tacrin, which was hepatotoxic, why they pulled it off the market. 200 micrograms twice a day. Safe, effective, and I'd like to throw out there, even in the kind of nutraceutical world, it's very common to use the isolated huperzine A as a single compound, single molecule. But imagine how much better it might be if you put it with the whole fern, the whole moss, and you put it all together. You had that whole matrix with a concentration of maybe it's 50% huperzine A or 20%. Question? No, you can combine it. Absolutely. Yep, totally. The question was, for patients who are taking acetylcholinesterase inhibitors, can you combine them with this plant compound? And yes, the answer is yes. And obviously, I'd rather have you combine it with the whole plant. Like I said, it's a Chinese herb. We can get a hold of the whole plant. And a couple of years ago, I was talking about what my dream was. Ultimately, was to build a farm where I could have any plant that I wanted on that farm. And if that plant made a compound in response to an ecological pressure, like let's say a fungus that was, like let's take resveratrol, right? So grapes make resveratrol to try to eliminate funguses that would, destroy their life. So they make a lot of resveratrol. So I would grow grapes that had a lot of fungus on them. I'd apply the fungus and they would make resveratrol. And I'd have high resveratrol grapes. And there would still be a matrix of compounds together. But that's really intensive and hard to do. And I don't quite have the resources to build that farm yet. So the other way to go is to take a bunch of that material, grow it, and extract the resveratrol from it, and then recombine it in some percentage with the whole plant. So I still get the matrix effect, but not quite like it's all married together perfectly. Yes. Yeah. It's a Qian Ceng Ta. Let me see. It's kind of, normally we do a talk like this, especially with all this information. It's good to have your notes, but I don't have any notes. So I'm just winging it. Let's see. I'll get this for you. Okay, you know what? Let's just keep going. I'll get you at the break. How about that? How much time do we have before break? 5 minutes? Okay. We're almost to halfway. That's great. So I just want to mention antipsychotics. They just don't work. They're not good in this group of people. They've been basically banned in treating patients who have any of these kind of neurocognitive decline disorders because it just gets worse. Let's mention some of the other drugs while we're doing our pharmacy stuff. Blood pressure meds, beta blockers. There's been some evidence that beta blockers can help slow the progression of AD, and I think that's probably because they block the adrenergic receptors, right? They stop the stress response. They calm the heart down. They stop that sort of, oh my God, they kind of chill it out. Of course, it's like a governor. You can't get excited about anything like the way you used to, but they might have an effect on limiting that amount of excitation and neurotoxicity that might occur under the normal influence of stress. But that's going to remains to be seen, along with the ACE inhibitors as well. Angiotensin converting enzyme inhibitors. They inhibit the enzyme that converts angiotensin. Anti-HCl agents, the proton pump inhibitors and the histamine 2 antagonists, I'm going to hit those on a slide. I'm going to hit the benzodiazepines. And then the SSRIs, I'll just mention, brain plasticity is a really important thing to think about. And SSRIs, selective serotonin reuptake inhibitors, that gets into that region of the brain that's super important, our serotonin, our meaning, our purpose center. And in people who are depressed, who don't have much serotonin, giving them serotonin receptor inhibitors, these reuptake inhibitors, when you block the reuptake of serotonin and make serotonin more available, what happens is the plasticity of the brain responds by changing the receptor relationship to the environment in the synapse. That's dangerous territory. It's like building a scaffolding around your neurological structure that holds it up. When you pull that scaffolding out, there's nothing there. That foundation that you built around it is not strong. So you've got to be very careful with these drugs. And what happens is, down the road, we see that all these drugs cause a lot of problems for people in the realm of neurocognition. And SSRIs are a scary one for me. I know that in some people who are really suffering, they can be very helpful. Again, I like to use plants, and I love to combine Hypericum perforatum with serotonin reuptake inhibitors. You do it in a careful and thoughtful way, where they're bringing in one and reducing the other. If you look at the research on Hypericum and comparisons with SSRIs, it's better than them in many studies. If you look at the studies that showed that Hypericum was not effective, analyze the study and see where the problems are. So proton pump inhibitors, one of the most widely prescribed drugs in the world and one of the hardest drugs to get people off of. You guys had people come in, they have acid regurg and they're having heartburn and they're taking, proton pump inhibitors. It's like, okay, well, they're like, I want to stop this drug. I just don't think it's doing anything. I think it's not really helping me in the long run. So what do? You're like, okay, well, let's start to get you off of it. Sometimes it can be really, really hard to get people off of it because it blocks the whole functioning, the whole connection, the brain signaling between hydrochloric acid and your food and what you're eating and how you're digesting it. And then hydrochloric acid is a signal for gastrin, for enterokinase, for the making of bicarbonate buffer, for bile, for pancreatic enzymes. It inhibits all of that, which is all connected to your brain and function and cognition. So now suddenly you just removed yourself from the digestive process. It's very, very hard on the body and hard to change when people are on it. Again, these were approved for 2 weeks. The black box, the label on these drugs says don't give this to people long term. It says they're supposed to be used for up to 2 weeks. How many patients come into your office and they've been on these for years? Right? They're over the counter. It's ridiculous. Increased risk of dementia. Absolutely. More studies are coming out right now. Same thing with histamine antagonists. They're blocking the histamine, which signals the hydrochloric acid production in the parietal cells. Benzodiazepines. Anybody like these drugs? God, they're just freaking terrible. Valium, Ativan, Xanax. Again, in the emergency room, these are awesome drugs to have. Man, when people are psychotic, they're out of their minds. You got to sedate them, or you got to put someone to sleep because they're going to go crazy. These drugs are great for that. Again, this is not a solution to get somebody to sleep long term. Taking Valium every night, I mean, I always think of sleep as this place. Like, sleep is this place where you enter this realm where you're sort of half in reality, half out. Then you go into a deeper place where you're dreaming and you're working out your emotional stuff. And then you go to the deepest, deepest place where you're gone. There's no memory of anything. And in that moment, I feel like my whole ego experience has left my body, and my body's actually able to do whatever it does. It's like my cells can act locally and think globally. They can do their thing and not have me involved in whatever I'm thinking about. That moment, it's like the meditative moment. That moment is so important. I feel like that is the key. And if you watch cycles in sleep, it's like we go through this cycle in sleep where we deep sleep and then we come to the surface. That's how we go. With these drugs, you're never going to go below what, if this is the range and you can get down to the bottom, these drugs might get you to here. But they're going to hold you here. You never get to that place. People never get to restore and rebuild like they would with real sleep on these medications. And what we're seeing now is that the longer you use them, the greater the risk. This is almost 2,000 people, right? First diagnosis of Alzheimer's, followed up for 6 years, matched with 7,000 controls. Clear association with Alzheimer's. Public health concern. Well-documented effects. So if somebody's going to use these for the short term because they're in an acute situation and they could get helped by stimulating the benzodiazepine receptor, then use a shorter acting drug, and you can look up the list, and they show you, you can look at Wikipedia, it'll tell you which ones are shorter and longer acting, and don't have them take it for a very long time. Very simple. So why don't we just get people exercising? We're almost there, Doug, we're like a minute. If we could just get people exercising, they're going to feel better. They're going to make their own natural receptor stimulation for GABA. The most persuasive evidence to date on exercise is for high-intensity interventions. And I am such a big proponent, even in people who are old and frail, to push your body. The body answers your push by making you more. That's why when guys go to the gym and work out, they get bigger. That's why when women go to the gym and work out, they get bigger. It's the same. And I don't know if you guys have noticed, but like there's a major gender shift happening in this world, in this culture, right? The lines between what women and men are supposed to do and how they're supposed to look and how they're supposed to be are starting to fall apart. We're starting to get beyond our structures of pattern response of the woman is at knitting or doing the thing and the man is out hunting, and that's all breaking down. And as I see people, as I see great tennis players like Venus and her sister, Serena, I'm like, these people are amazing. You see, women are pushing their bodies, and their bodies are responding. And as they push their bodies and their bodies respond, their offspring will have more advantages through those single nucleotide polymorphisms that they alter through their actions, through their life. We are going to come to a place, I believe, where women and men are much more equal in that regard. And I'm making this point because we can push our bodies to make more of us. One of the best ways to build testosterone is to push your body to that point where you get to muscle fatigue. Break down, I can't do it anymore. Not just I did it 12 reps, it was fun, sitting in front of the TV on my little stationary bike. That's not what I'm talking about. I'm talking about push yourself. And the body responds by giving you that burst of what you need. Improves mood. So last thing on AD here, just Mediterranean diet, music, art, drama. There's really been no conclusive evidence for exercise. I did a bunch of research on this, but I think there's a threshold at which you have to get to that point where you push yourself to that fatigue point. You have to make the body really want it and work. You can't just do walking. People say, I walk every day. I work in the garden. Well, I'm sorry, but it's not going to probably be enough. We need to push our body, get to that point where you actually feel burning, where you can't get enough oxygen to make happen what you're asking it to do. And the other thing is, when we combine exercise, especially that high-intensity exercise, and again, pulsatile. So if someone's walking, walk and then sprint for a little bit. Run for a little bit. Do a couple squats. Do some lunges. Just do some of this. Push your body. Do it slow. You'll get amazing results. But if you combine it with the things we're going to talk about in a few minutes after we come back from break, with Rapunticum carthamoides and Mumi, you combine it with Panax ginseng, and you start looking at all these plants that will help enhance our anabolic response, we're going to see really profound results.